It's a subject that I've been very interested in for a couple of decades. And after doing some due diligence, found the world's expert on hormesis, who is our next speaker. Dr. Ed Calabrese is the professor of toxicology at the University of Massachusetts, their School of Public Health and Health Sciences at Amherst. Dr. Calabrese has researched extensively in the area of host factors affecting susceptibility to pollutants, and is the author of over 825 papers in scholarly journals, as well as over 10 textbooks. He has been a member of the United States National Academy of Sciences and the NATO countries' safe drinking water committees. He also serves on the board of the Scientific Counselors for the Agency for Toxic Substances and Disease Registry, the ATSDR. Dr. Calabrese also serves as chairman of the Biological Effects of Low-Level Exposures and director of the Northeast Regional Environmental Public Health Center at the University of Massachusetts. Over the past 25 years, he has focused his research into understanding the nature of the dose response in low-dose zone and underlying adaptive explanatory mechanisms. Please give a warm Academy welcome to Dr. Ed Calabrese.
In terms of the hormesis concept, and I'll get into some definition a little bit in about a minute or two, but low doses of numerous things, numerous stresses, things we kind of take for granted, things like exercising, you probably heard of intermittent fasting, hypoxia, heat, cold, ionizing radiation, non-ionizing radiation, electricity, toxins, drugs, chemicals. All these things can stimulate a wide range of adaptive responses. These induced adaptive responses, they have the potential to profoundly affect the success of many public health practices and medical interventions. Some of these you're involved with, such as surgeries, chemo and radiation therapies, and for numerous disorders, really affecting susceptibility and damage from heart attack, stroke, shock, brain traumatic damage, organ transplants. Now, stresses that can trigger adaptive responses also offer ways to enhance healthy aging. They can improve human performance and prevent damage to tissues exposed later to a wide range of injurious stresses. And the previous talk, it was the term preconditioning came up quite a bit, and how it's used in therapy, and how he used it in therapy I was totally unaware of. And so this was actually very interesting to me in many ways. And so at the center of this adaptive response, things that we're pretty familiar with, in my opinion, is the concept of hormesis. And hormesis is a biphasic dose-response relationship that mediates processes by which human adaptation and performance can be improved. So what is hormesis? How can it be used to achieve these public health and medical advances? And when and how should hormesis be taught to undergraduates and undergraduates and medical students? Well, in my opinion, it's kind of hormesis by Calabrese. Hormesis is a dose-response phenomenon, and it's characterized by a low-dose stimulation and a high-dose inhibition. It's a non-monotonic biphasic dose response, but it's not just any type of dose response. It's a very specific type of dose response. It has very specific quantitative features that we'll talk about. Now, in the course of your life in medicine and toxicology, it's been really dominated, the field's been dominated by two dose-response relationships. It's kind of all I taught the first 25 years as a faculty member, was the threshold model. Now you can have a dose response here with a low dose going out to a higher dose and your response here. The line going down the middle is a normalized control group. So this is like your 100% line. And the threshold is this, no action really going on, and then bingo, you hit some sort of magic dose and biology takes over and you see an effect. So we'll call that the threshold model, here being the threshold, below the threshold, not expecting to see any biological treatment activity. And the US EPA and their regulatory actions and the FDA, they assume that all non-carcinogens, for the most part, follow a threshold dose-response relationship. So the heavy metals, things that you might be interested in, for example, mercury, some of its neurotoxicity, even if it's highly toxic, it's still supposed to act by a threshold model for the most part. That's how it's seen by the regulatory agencies. However, in terms of carcinogens, EPA and the FDA, and all countries across the globe, they assume that carcinogens do not show a threshold, but show a linear relationship. This means that there really is no safe exposure to a carcinogen. It means that the exposure, or the risk, is proportionate to dose. So you could take this to a logical extreme, and you could say, well, it's generally accepted that cigarette smoking increases the risk of developing various types of cancer, such as lung cancer. And if you really wanted to extrapolate, you could say, well, even a single puff could essentially increase your risk if you wanted to take it to a logical extreme. You can say even a single ionization from radiation could do the same thing. The hormetic model is really a third model, and you can see it's really very different than the other two. At high doses, they're pretty much the same. They're showing that things go, you might say, towards these adverse effects as the dose is increased. But you can see this hormetic model schematically is showing that that risk is actually reduced at low doses. Okay, that's the radical part of this. Okay, the high dose isn't the radical part. This is saying things, for example, such as radiation, such as any toxin that you want to make. You could even say, dioxin. You could say mercury. You could say lead. You could say any type of pesticide. I'm claiming that it fits into this biphasic dose-response relationship. But something goes on differently at low than it does at high. Doesn't mean that the effect is necessarily beneficial, because I'll show you some examples where it doesn't. But you can see that this clashes with the two main paradigms that are out there. And essentially, is what I'll be proclaiming anyways, is that this is the dominant paradigm. I'm going to be proclaiming that the regulatory agencies are wrong, and all the regulations are based upon faulty understandings of the dose-response relationship. And I'll try to share with you why I think that's the case. Okay, now I have created 4 very large Hormesis databases for different reasons, trying to answer different questions. And one thing that you realize when you look at really tens of thousands of examples of these, and it struck me rather that I didn't expect, was that regardless of the agent, regardless of the endpoint, regardless of the model, you have generally similar quantitative features with respect to the amplitude, the height, and the width of this low-dose stimulatory response. Now this occurs, these biphasic dose responses will occur either through a direct stimulation, expose the agent or apply the agent, and you'll see this low-dose stimulation, or it could initially induce some toxicity, and then you'll see an overcompensation response. Now regardless of the you might say that the overall mechanism or the mode of action, whether it's a direct stimulation or an overcompensation reaction, the weird thing, something I didn't expect, was that the quantitative features of that dose response remain the same. That's going to be an important thing, so remember that, because it's going to be very important from an evolutionary point of view, from a pharmaceutical point of view, and public health point of view. Now, in terms of how to interpret this dose response, most people, and the big issues with regulatory agencies is people argue and debate, is this low-dose stimulation beneficial, harmful? In my opinion, that really shouldn't be part of the definition of hormesis. Hormesis it's not about benefit and harm. It has to get to that, but it's really about essentially hormesis as a dose-response relationship. We will eventually try to interpret a specific dose response, whether that low dose is harmful or beneficial, once we really get into it. And I'll give you an example here of where that low-dose stimulation actually is not beneficial, not to patients anyways. This is a schematic that's taken from a very large public database, the National Cancer Institute database, where many thousands of potential chemotherapeutic agents for treating cancer are available, where they have tested them over a broad range of doses. And most people in the pharmaceutical world, they're interested in anti-cancer drugs. They want the drug to kill the tumor cell. And so they're interested in high dose effects. And typically you'll see at high doses you have an inhibition of growth, and you'll have the beginnings of the mortality of those particular cancer cells. However, most of those people aren't interested in what goes on at the lower dose ends, but that's what I am. And we went and did a study of essentially many thousands of those, essentially the whole database. And we found that basically all those drugs were toxic in high doses, but if you really looked at what they did at low doses, they tended to stimulate the tumors and the tumor cells to proliferate. So drugs that could be used to treat the cancer cell at high doses actually proliferate those tumor cells at low doses. And so that to me is something that you don't want. So, but in my opinion, this was also a hormetic dose response. It's not a beneficial response. Maybe beneficial to the tumor cell, but it's not beneficial to the patient. Now, if you go back into the literature, you find the same type of thing tied into antibiotics, things like penicillin, streptomycin, and so forth. If you could go back into the literature, and I have put this together, you'll see the same type of dose response where at high doses, the antibiotic may be killing the harmful bacteria, but actually at low doses, it actually can stimulate its proliferation. It's a very interesting situation. Now, I'll give you an opposite situation where the low dose could be beneficial. In the world of memory, there's an awful lot of drugs that have been developed to try to improve our memory. Many of them have been tested in rodent models. And if you look at hundreds of these examples, you find that these drugs can often increase the memory of the rodent in the various studies that they do. And at low doses, you see this low dose stimulation, but at higher doses, it actually impedes the memory. And the interesting thing is that the quantitative features of this enhancement of memory, it's really the same quantitative features that would be shown in the previous slide with the anti-tumor drugs. It follows the same type of dose-response relationship with a totally different model and a totally different endpoint, totally different framework. Yet the dose-response features are the same, and in my opinion, they're basically hormetic dose responses. The two fields never talk to each other. They wouldn't even know that there's same dose responses occurring in two different fields. Their literature, their training, everything is really different, even if there are basic biology people there. I'll get into this a little bit more in a second, but I study dose responses really by really looking at about 5 different things. We look at patterns, we look at statistical significance, replication of findings, the mechanism, and we look at simulation studies, and I'll talk about these in their due course. Now, how did I first get involved with this sort of a thing? And it was really by accident. I was an undergraduate many, many years ago, and I was taking a plant physiology class. And in this class, we had to do like 30 or 40 different experiments in the lab in a greenhouse. And we were going to be treating these peppermint plants with a synthetic plant inhibitor. And so we went out and did this exercise for the class, and didn't think too much about it because there were so many other little experiments to do. The professor comes back at the end of, towards the end of the semester and says, something's going on wrong with this experiment. The plants are actually being stimulated, and they're not supposed to do this, and we do this little exercise every year. It's never shown this stimulation. You people have screwed up somehow, and I just don't know how you screwed up, but you did. And he said, and so I'd like to find out why. Now, if somebody would like, it's too late in the semester to figure it out, but if anybody will come back and kind of work with me, maybe we can figure out why these plants were stimulated. Well, you know, the story is, of course, is that I was the only one who went back, and essentially this looked at my notebook and looked at what he wanted us to do, and it appeared as though that my group, probably me, made up our stock solution wrong, and by essentially off by a factor of 10. too dilute. And so when we did the experiment over again with his high doses and our low doses, believe it or not, we got a low dose stimulation and a high dose inhibition. We got exactly what he said we would get at the doses he said, but still our low dose stimulation came up. And so we kind of had an answer to this little mystery. And then he said, well, you have to repeat that study. And he said, in science, you have to replicate your work. So I went back and I did the experiment kind of over again. And then he says, I said, I replicate. He says, it's not strong enough. You don't have enough doses in that low dose zone. The stimulation was only 30 or 40%. You have to go back there and have more doses, more plants. You have to replicate it again. I went through literally 10 replications of this particular experiment. He was never satisfied. He would never believe it. It needed to be, and I didn't have his insight as to what the significance of this all was, because actually what I'm going to show you is, see that little bump up that I showed you before, that little bump, that's only about 30 to 60% above the control group. That's not a big bump. That could be easily, if you have a lot of experience in the lab, easily variability. You could see a result. If it conforms to your belief system, then you can believe it. Okay, and that's not the way to do it. You actually have to have strong study designs. You have to, fairly test a hypothesis. You have to repeat that hypothesis testing over and over and over, especially if it's a modest increase. And then ultimately, what happened in my situation, after really 10 times, I went to him and said, I've had it. I'm quitting. I'm not doing anymore. I mean, you blew my mind mostly. And so he said, I agree. Finally, okay. He said, but you created a new idea. He says, maybe what's happening with these plants is maybe the chemical is interacting with some microbes in the soil, and maybe they're creating a new chemical form. So let's try this same study, but do it in hydroponics away from the soil. I said, that's actually a pretty good idea. so I went and did that study, learned how to do hydroponics, and replicated about 6 times and got the same biphasic dose response, and thank God graduated. And I never knew the term hormesis, but it was my introduction to a biphasic dose response. And that's how I got into it. And then fast forward 20 years, I was a insecticide toxicology person, really. And I see a conference being held in Oakland, California, on this concept called radiation hormesis, and they're talking about biphasic dose responses. And it must have been a boring day or so, because I decided to call the guy up who was running the conference, and I told him the story I told you, that I think maybe he was interested in low dose of radiation could reduce the incidence of cancer. And I said, biphasic dose responses are biphasic dose responses, and biology is biology. These have to be related somehow. So I called them up and I told them my story, and they invited me out to California to give a talk, not on chemical hormesis, that I didn't know anything about at the time, but on, not on radiation hormesis, but on chemical hormesis, so I could come up with some different examples. And so the first conference on radiation hormesis was really held back in 1985 in August, in Oakland, and I never made it to the conference because my wife was expecting our second kid within 10 days of the conference date, so I actually never even showed up to the conference. I sent a graduate student out there, but nonetheless, that's how it all started for me. So now I have these thousands of examples in multiple databases indicative of hormesis using very rigorous criteria, and so I've been putting these together during the 1990s to the present, and the concept of hormesis had never made it into the textbooks. I mean, I've been at UMass as a faculty member since '76. And taught from '76 into the '90s and never mentioned the term hormesis. Everything was always linear, threshold, linear and threshold. Stuff it down their throat, stuff down my throat, linear and threshold. And now I'm beginning to challenge my own belief system on this. So in my opinion, based upon looking at all these examples, I'm finding that this hormesis thing is a very general phenomenon. It's really independent of model. This is a very interesting thing. What you see this will occur in plants, it's occur in bacteria, yeast and fungi. It'll occur in fish. It'll occur all the way up to humans. It'll occur at the cell level, at the organ level, and the organism level. That's also interesting too, at different levels of biological organization. And the weird thing is that you have the same quantitative features of the dose response regardless of the model, regardless of the agent, regardless of the endpoint. And this might further surprise you, and we've proven this recently, and independent of the actual mechanism. Doesn't make any difference which receptors involved, which pathways are involved, how they talk or don't talk to each other. The quantitative features of the dose response come out the same. And how is that possible? How is all that possible? What's going on biologically? Why would that be conserved evolutionarily? And you'll see that the dose response is modest. It's only 30 to 60% greater than the control, usually not more than 100%. The width can be more variable, and this needs to be studied. But generally speaking, in the experimental aspects where you have a lot more genetic similarity, it tends to be within about 20-fold of where the threshold is of what I'm calling a Noel term. This is the standard schematic that I show. You have a dose-response relationship with a low dose going out high, and then the response is on the vertical. And you see this low dose stimulation and this high dose inhibition. It only goes up by about 30 to 60% above the control. And you have to experiment with this. As my old advisor forced me to do, you have to have a lot of doses in this low dose zone. You have to replicate that to see that it is reproducible and real. Too much experience in lab stuff to know that you could be easily fooled to think something is real and it's actually variation. And you can't make a career on that. To actually prove things. I'm showing you here normal dose response for a normal individual and a high-risk group. Back in 2002, I was asked to write an article for a publication called the Annual Review of Public Health, and it's kind of a prestigious series in different areas. If you're asked to write an article for that kind of a series, you're supposed to be an expert. And so I wrote this article on hormesis and public health. And so as it turns out, I've never seen this done, but one of the members of the editorial board decided to challenge my annual review paper with a criticism of it written in the same issue. And usually, it's never happened before from what I can tell. So there was... it should have been one of these life's embarrassing moments where some of you have your prestigious article and you're the other guy who's speaking, and then somebody on the editorial board says, really challenges you. But what his challenge was actually, it was actually a good idea, his challenge, because in the write-up, at least in public health and environmental standards, you're trying to protect the public, general public, but you're also trying to protect people who are at increased risk, whether they're bronchitics, asthmatics, or people who have different types of predisposed conditions. And we didn't do that. And so I said, well, does hormesis take place in those who are at compromised health? And so we went back into our databases and we did this and did that, and we found lots of examples that we had previously kind of ignored or just weren't taking it too seriously. And we went back and we traced it all out, and we found that the high-risk people showed the exact same type of dose response as the normal, same quantitative features, but shifted to the left on the dose curve. They just responded at a lower dose. So his criticism of our work was really a good thing, and it led us to do a better job. Should have done a better job the first time, but it was good. If you go into the world of pharmacology, you find, regardless of all these different, basically major agonists that kind of control our life, whether we're talking about dopamine or estrogen or spermine or prolactin or adenosine, all these things that have major factors within our lives, our cells, all of these that affect any endpoint that's significant to us, they all act by biphasic dose-response relationships, all with the same quantitative features of the dose response. Your entire biology is biphasic, and low doses respond differently than high doses. Now, we've looked at mechanisms, and I have to tell you, this is interesting. Just a little snippet. Back in 1999 or so, I submitted my professional society is the Society of Toxicology, and I submitted six papers to the Journal of Toxicological Sciences, and I got 6 papers that had decent reviews, and all 6 got rejected. I got 6 papers rejected in very short order. And so I said, God, what is going on here? So I called up my grant manager, who was from the U.S. Air Force, and I told him my experience. And he said, Ed, if you want to get this grant renewed, you're going to have to get papers in Tox Sciences and other ones that look just like it, because we want the top level journals. Your stuff has to get in the top journals. And if you can't do it with this project, you're not going to get renewed. So I said, what am I going to do here to kind of save this thing? So what I did was I said, what the hell? I said, you know what I'm going to do? I called my grant manager up. I said, I need $50,000 more than what you're giving me. And I want to invite that guy who has rejected me 6 times in a row, and then 7 other people who made their name on low-dose linearity. And I want to debate them in front of 300 people in a 3-day conference, and we'll see what's left standing. And so the first day, I will give a 6-hour, like Fidel Castro talk on hormesis, everything I know. And we did 32 posters as well, just us. And then the second day, all the other people would come in, and they would more or less pummel me. And then the third day, we'd see what's left standing. And I said, well, I'm either going to survive this or just get, flare out at this point in time. And so 300 people showed up in January of 2000 at a conference, and to watch the hormesis showdown at the OK Corral. And so I went out there and I gave the best and only 6-hour talk I could ever give on hormesis. Did the 31 posters, just our group, and 2 is enough to drive you crazy. And so I came home and my wife said, well, how'd you do? And I said, I had the audience. It was the best talk I've ever given on hormesis. I've got them. This is over. And so I showed up the next day, and the statistician got up, and he says, see, I had sent these people 150 papers, the best papers that I knew on hormesis, 9 months before to prepare for it. So they should have read it and all this. He got up and said, I didn't look at the 150 papers much at all. I'm just going to assume that, in fact, that the hormesis is real. He said, but I can tell you, I have a problem with what Calabrese is doing. He said, Calabrese has thousands of examples of hormesis, but he's got a flaw in his database. And the flaw is that he has lots of cases, examples of hormesis, but he doesn't have, he had examples based on evaluative criteria, but he didn't have any entry criteria. It would be like a baseball person coming up and saying, I had a great season, I had 150 base hits, and the person would say, well, how many times did you get up? Well, gee, I don't know. And so you can't figure out the guy's batting average. It'd be like, I got 13 right on the exam, but how many questions did they ask? I got 13 right on the exam. Well, they asked you 1,000 questions, or did they ask you 13 questions? So I had to admit in front of the audience and my grant manager that he was right, that I could not figure out a frequency. And I had 5,000, 10,000 examples of 1 million or 5,000, and I couldn't know. So I started losing ground in the debate. and then the guy from Tox Sciences got up there and says, I don't care about the frequency of hormesis in the literature. In toxicology today, we worship at the altar of mechanism. And until he can figure out a mechanism, he's not getting into my journal. And then the next guy got up, and the next one got up, and the next one got up. And I kept getting pummeled, actually. And so I came home after the second day, and my wife said, how'd you do? And I said, remember the movie Rocky 1? And I said, I'm the guy who was fighting Apollo Creed. And I didn't, my big swollen eyes and, bloody lip. But I was still standing. I have to admit, I was standing, I was wobbly, but I was still standing. And so I learned that, just like I did with this guy from the annual review, I learned that was a really powerful learning moment for me, to go up there and get your lumps in front of your graduate students and your, and your grant manager. And what it did, though, is it allowed me to go back now and to build a proper, a new database that would have a priori entry and evaluative criteria. And so I went back and I did that, and I tried to learn from my past mistakes. And I ended up ultimately getting that next paper into Tox Sciences with the first ever hormesis frequency within the literature. Now, so I have So what I did was I was able to answer that statistician's question on the frequency, and we came up with about 40%, which is actually pretty darn good with very strict criteria. And then I was able to go back in and try to answer this toxicology guy's thing in terms of the mechanism, and we published actually a paper with 400 different toxicological mechanisms for hormesis in one of the leading journals, and put that on the map. So the critics that you have, if they're fair-minded, they can be the most helpful people in the world to you. I mean, really, you learn a lot more from losing if you really reflect on how you could improve. And so I'd like to thank these people for what they did. So basically, regardless of mechanism, I'll show you more examples, doesn't make any difference whether the mechanism is receptor-based, signaling pathway, non-receptor mediated, direct, or compensatory. The quantitative features are the same, and we'll look at that and why. Now, the hormetic responses that I like to look at and emphasize are integrative responses. And what's an integrative response? An integrative response to me is something that really serves life. For example, cell proliferation, fecundity, tissue repair, behavior, learning, disease incidence, resistance to disease, DNA damage, tumor incidence, how long do you live, the quality of your life, these things that speak to health. I believe that this is what hormesis is actually speaking to, and it has many mechanisms that lead to each of these. Now I'm going to show you some examples now of what a hormetic dose response looks like. And I have, as I said, I have really many tens of thousands of these. The examples that I have selected for you are mostly to show the range and diversity of endpoints. So this over here is the effects of ethanol on social behavior in rats. And you can see at high doses, the ethanol basically impedes or slows down their social interaction, but at low doses, it flips to the other side of that 100% control line. But down here you have the dose of ethanol, over here you have the response, but the control group is 100. And so you can see it's flipping over, and at maximum is about 50% more than the control. This over here is an example of males and female fruit flies and looking at longevity. And you can see at high doses, the methanol, which is a toxic substance, is very damaging to their longevity. But at low doses, they live longer. And this is a very, very reproducible finding throughout the literature. This over here is this substance, allicin, which is found in garlic. And you have a dose response. I mean, you can see it's looking at a survival of primary cultured hippocampal neurons from a Wistar rat. And at high doses, this allicin is toxic to those cells. But at low doses, again, it is enhancing of survival. Here you have sodium arsenate, and it's looking at lymphocyte stimulation. And at high doses, it clearly is inhibitory, but it flips to the other side and goes up, to about 50 to 60% at max. Methyl mercury, which is the most toxic thing that EPA actually regulates. This is being tested now in a neural cell line. And at high doses, it is harmful. But it's interesting that at low doses in this neural cell line, it actually enhances survival. It's very, very interesting, very weird biology here when you really take a look at low doses and what could be going on. And they're always modest. They're always modest. And why you want to have replication in many doses is you want to see how consistent that response is. Over here, we're looking at nicotine. And nicotine, if you're a heavy smoker, the epidemiology suggests that it's associated with osteoporosis. It can cause bone damage. Now, but you're going to see over here in this particular study with human osteoblast-like cells that, in fact, you actually have an increase in the proliferation of those bone cells at low doses, but it's inhibited at high. Now, the little blobs over here are attempts at mechanistic studies where they have been able to block this stimulation by blocking a particular receptor. So you kind of know what receptor is mediating that response. They tried to block the same receptor here. It had no impact. So that receptor is not mediating the inhibition, but it's mediating the stimulation. But the point is that something's going on at low doses that's not going on at high. And if you just try to linearly extrapolate back for the linear dose response or a threshold, you'd never see this. Over here, this is arsenic, and it's looking at the effects of arsenic on cell proliferation in the human embryo lung fibroblast cells. And again, you have a biphasic dose response. All these blobs are, what I'm just trying to represent to you, are very detailed efforts to try to figure out a receptor or a pathway that's mediating the stimulation, or a receptor or pathway that is mediating the inhibition. And without going into the details of these here, just know that there's a lot of mechanistic research that is going on. This over here is a different sort of a thing. This is now looking at a drug that is related to seizures. And I'm not a seizure person, but I somehow fell across some papers on seizures. And there are different models for seizures. And in this particular case, what you are looking at is when it takes more drug to induce a seizure, then the drug has potential for therapeutic application. Here we're looking at morphine in a PTZ seizure model in an animal, and it's requiring about 25% more morphine, requiring 25% more PTZ to induce the seizure at this particular concentration of morphine. I looked at that and I said, wow, I said, in this seizure model, it's a biphasic model. It conforms to a hormesis. That's hormetic dose-response relationship. And now I found about nearly 100 examples of this in the preclinical, essentially epileptic literature. And it's essentially all the medications that they produce are all hormetic dose-response medications. This over here is one that is looking at anxiolytic drugs, drugs that will reduce your anxiety. And they study these in mice and rats, and you study them in mice and rats by seeing if you can get the mouse and rat to do something it doesn't want to do, and usually they like to hang out where it's dark, and if you can get them to spend more time in a lighted zone, then that's a sign that they can possibly overcome their anxiety. And so here they're measuring the amount of time spent in the lighted zone, and it shows a beautiful hormetic stimulation. In high doses, they become even more anxious. And essentially, I found about 150 different anxiolytic drugs. Essentially, they all show this biphasic dose response. The entire pharmaceutical industry that's built on anxiolytic drugs and anti-seizure drugs, it's all based on hormesis. Now, this over here, if you had red wine last night, we're looking at resveratrol. And resveratrol is studied here cell-wise, looking at its capacity to reduce myocardial infarct damage and cardiomyocyte apoptosis. And you can see that at low doses you have less damage, but at higher doses the resveratrol actually increases the damage. I think I went back, yeah. Now, as a follow-up to the preconditioning we heard earlier, I'm going to give a couple of examples. Preconditioning, as you heard, is when you give a low stress to an organism, and you wait about a day or two, and you give a big stress to the organism, and that low stress oftentimes can protect against the high stress. And I'll relate this back to hormesis in a second. Here, what we're doing is we're giving a low dose of, in this particular case, it's hydrogen peroxide, and it goes from basically 0.05 out to 5. And then we're going to wait a day or so, and then the investigators then gave a high dose, 50. And that's the damage that is normalized to 100% here. If you got the pre-dose, it reduces that damage from that 50 by about 50% in this optimal zone. And then at the high dose, it actually starts to get worse. It follows a beautiful J-shaped dose response. It's a very hormetic type of response. Over here, we have preconditioning with nitrite that is given prior to inducing a heart attack. And you can see here that you reduce the damage by about 50%, and then it goes up. The preconditioning actually protects. And I'm going to go back to the other one in a second. How that all came to be was, in fact, back in 1986, investigators at Duke were trying to induce angina pain in a dog with altering or really blocking the flow of blood to the heart. And then they wanted to wait a day and then induce a massive myocardial infarction in the dog and see if that changed the damage. And so they did that, and the dogs that were preconditioned with the hypoxic insult, so to speak, they had damage reduced by about 80%. It was actually an amazing finding. They were able to reproduce their work. They were able to get other people to reproduce their work with mice, rats, gerbils, guinea pigs, up to sheep and so forth, and other species as well. And they were actually able to, and they called the term preconditioning. So in the biomedical world, the term preconditioning really starts around 86. And the paper that showed this has been cited about 7,000 times now in the Web of Science. And so it's now been expanded to really in so many different ways. Okay, I think I'm going in the wrong direction here. Yeah, and this is another example of preconditioning, and it's a drug in which you actually, in this particular case, I'm showing you two dose responses. This is just a direct effect of this drug on cell proliferation, cell survival. And then this is essentially when you damage you're going to give a high dose, but you're going to precondition with the same dose but at lower doses. And you can see that if you precondition, you can reverse a lot of the damage that will have been induced. You can reduce survival by about 50% with a very high dose. But if you precondition, you can prevent some of that damage. And this begins to make you think about its role in evolution. Now, what's actually happening here is that why I relate preconditioning here is that I went back into the literature and I looked at hundreds and hundreds and hundreds of papers, thousands really, on preconditioning. And I found that when they had a broad dose response relationship, so that you could evaluate the preconditioning dose, that it conformed beautifully to a hormetic dose response relationship. That preconditioning was actually hormesis. Pretty amazing. And the quantitative features of preconditioning were actually the same quantitative features that I've just showed you with hormesis. So we published a couple papers 2 years ago in Pharmacological Research entitled, Preconditioning is Hormesis, with all the documentation. And this relates back to the opening salvo, and that is that if when you are being exposed to low doses of stresses, how does your body respond? And really what your body is going to be doing is upregulating adaptive mechanisms that may protect you against a subsequent harmful event. Now that can protect you against damage from a heart attack or damage from stroke, damage from a brain traumatic injury. There was, I mentioned to you the issue of the dogs and the heart attack. Investigator, actually at UMass Medical School, and now she's at Wayne State, she followed this further, and she wanted to find a non-surgical way that you could protect the heart. So what she did, this is very interesting, you probably know about this, but she used a blood pressure cuff. And it was kind of like in place of a tourniquet. And she put it on the hind leg of a rat. And she pumped it up, you might say, to 200 millimeters of mercury and held it there for 5 minutes. And then let it go for 5 minutes, and then did it again for 5 minutes, and did it 3 times, essentially. Waited a day, and then gave a massive heart attack to those rats. Those rats were as protected as the dog was. They had about 80% decrease in damage. They all essentially lived. Compared to those that didn't have that, they essentially died. And so this blood pressure cuff thing was actually pretty amazing, and it worked with a variety of animal models, And it was also very interesting because the cuff was also protecting the brain. The cuff would also protect the liver. If you pre-treated with the cuff and then you gave it carbon tetrachloride or dimethylnitrosamine, some liver toxin, you'd have a profound decrease in the onset of toxicity. You could do the same for a kidney damage as well. And when you played this thing out, your protection depended upon how many times and how long you used the cuff. Did you pump it up once? Did you pump it up twice? All the way up to 8 times. It followed actually a beautiful biphasic hormetic dose-response relationship. It's induction, in my opinion, of an adaptive response. The issue of hormesis is its capacity for generalization is really quite broad. Now I'm going to show you some examples of cancer. Now, cancer, as I was talking about this with respect to the linear dose-response relationship, but if the linear dose-response relationship, this is now looking at ionizing radiation and mouse lung adenomas. It's a very big study. Each one of these little, triangles and squares, they have about 500 to 700 mice in them. Now, the linear dose response should show a linear pattern right back to the control group. But you really don't see that. You see a biphasic dose response. It's a very hormetic dose response. You see a reduction in tumors at low dose, and they begin to get more tumors as the dose increases. They go down by about 30%. Very, very hormetic representation. I'll show you a Japanese study working with a mouse model that is predisposed to developing diabetes, a DD mouse. Now, this is showing you how well they're doing out at 92 weeks. Now, at 92 weeks, we have 40% of the control group being alive. We have 70% of the radiation-treated mice being alive. The level of radiation they're getting is about 60 to 70 times above background. I call that kind of a low dose, but it's still 70 times above background, so you might consider that a high dose. Now, I want to show you a representative animal from this. This is a representative from the control group. He looks like my dad. My dad lived about 95. This is how my dad looked at 95. He was well worn. He was getting quite frail, had lost a lot of hair. He really looked like, he had lived the life over 95 years, okay? This is the one that got treated with the radiation. Its fur is shiny, it's full, it's plump, and I said, give me what that animal's getting. I said, this is interesting. These individuals, you might think that lived 3 times longer than the control group, but it didn't. It lived about 20%, 30% longer. So somehow it seemed to me like it had healthy aging, but it didn't live out more than the 30% that you might have expected hormetically. But nonetheless, what I'm trying to represent to you is that 2 things. One is that low doses of radiation were probably beneficial to these animals. But over here, what's the biggest pollutant that we are exposed to in our lives? It's ourself. This creature was not exposed to anything harmful. It got a good diet, it was in a good environment, it was taken care of, had everything going for it. And look what happened to it after 92 weeks. It looks like a piece of crap, actually. And so the biggest polluter, if you want to call aging a pollution, is actually what we do to ourselves. It is aging. We are, you might say, oxidizing ourselves into obliteration. And really what the hormetic factor is to take control of your own life, and how can you actually build a biological shield that can slow down that rate of deterioration, give you a higher quality life, expand your other opportunities, a bit more like what we're seeing over here. This is the normal course of life. That's actually, we are our own biggest internal polluter. It's inside. And that's if you think you're doing well for yourself. EPA's approach to life, and I'm not really disagreeing with them. I disagree with them, but their view is lower is always better. Lower is always better. Well, okay, this animal had Laura is always better its whole damn life. So who won? It means you really can't, you can thank your regulatory agencies for protecting you. They didn't protect you from yourself or what we are. Okay, this is gamma rays, and it's looking at longevity in the house cricket, okay? And it responds just like the mouse and just like the human. Low dose, the high dose are harmful. Low doses actually and enhance its lifespan by about 50%. This over here is looking at mutation in the Drosophila. And now Drosophila is very significant because this was the model that Hermann Muller used to prove that radiation could cause mutation. The first person who ever showed that, and he won the Nobel Prize for what he did by studying high doses. If Muller had also studied low doses, he might have got 2 Nobel Prizes. Because you can see low doses are actually decreasing the occurrence of mutation. DDT is banned because it's a carcinogen and does other things, and it is a carcinogen at high doses. Japanese investigators decided to look at low doses, and in the same model that the US did to prove it was a carcinogen, but the Japanese showed using liver foci as a predictor of liver cancer that it in fact actually reduces liver cancer at low doses. And the biggest study ever to date, they call it the Mega Mouse study, that had 24,000 animals to try to prove what the nature of the dose response was in the low dose zone with this carcinogen 2-AAF, 2-acetylaminofluorine. High doses, it caused cancer to the bladder. But low doses actually didn't support this linear dose-response relationship. It was J-shaped. This had so many animals, actually. They had 8 different buildings and so forth for 8 different replicates. Each replicate showed a J-shaped dose response. It was a pretty convincing thing. So what is Hormesis actually indicating? In my opinion, this low-dose stimulation, it's actually different than high-dose toxicity. The low-dose stimulation, I believe, what it's really measuring is biological performance. It's not measuring toxicity. It determines how much a system can respond. I think what it's actually measuring is plasticity. How much flexibility do you have within the system? And this is why I think that it's so general across all of life. And that is that you have to figure why would evolution have selected for a certain quantitative limit to our capacity for plasticity? Because our capacity to adapt is limited. It's limited to, I think, the constraints as described by the hormetic dose response model. So when you take a drug and you're going to boost your memory, how much can you boost it? 30%, up to 60%. I get one drug that can boost me to 60%. I'm going to take another drug in their optimal zone and I'll double it. Maybe I'll get to 120%. I've seen those studies. They don't. They actually cap it at about 30 to 60%. You don't go outside plasticity. It's like redundant controls are keeping it capped. Why would we want to have redundant controls to cap how much we can do this? Reducing anxiety. When a drug company wants to reduce anxiety with these animals, they can't get it to go more than the 30 to 60%. That's actually very hard to prove epidemiologically because you study a broad population, you're trying to look at 30 to 60% increases. Those are pretty hard studies to prove because you have to make sure you have enough people, enough doses, take into account all this heterogeneity in the population. You still have to see this little tick up by 30 to 60%. Hormesis is a challenge. I study it in animal models where it's easier to study, where you can control all the variables and I can tick off this 30 to 60%. Human populations, so it's a different situation, but I believe that the maximum you can get out of what we have is essentially limited by plasticity, and Hormesis describes that, and it actually explains a lot. Okay, explains a lot. Okay, what are its applications? Well, broad spread applications. You know the whole world of big pharma, anxiolytic drugs, anti-seizure drugs, memory enhancement, osteoporosis, wound healing preparations. I didn't talk about wound healing today, but I have a gigantic set of stuff on wound healing and hormesis. It all conforms to this 30 to 60%. As far as I'm concerned, I've seen an awful lot of that. If you take a look at other types of things, there are a lot of applications of this to the world of organ transplant. You can pre-treat hormetically, optimal dose to enhance essentially organ transplants, to enhance surgical success, chemotherapy, and so forth, with a lot of different ways. Lifestyle factors can be, I call them little hormetogens or hormetins, but diet, intermittent fasting can be useful. What you're really trying to do in this is that if you have a low level of stress, a low level of stress induces adaptive responses. It'll protect you from a subsequent stress. And you can have, and there is such a thing as post-conditioning. We talked about preconditioning. It's very interesting. When you are inducing a heart attack, you can go in an animal model, induce that heart attack. You can go in right within a little time window afterwards, and this is really strange, but you can essentially do the same occlusion and release, occlusion and release, occlusion and release, but do it in seconds, not in minutes. And you can get the same protection that you would get if you preconditioned. It's very, very interesting. The quantitative features are really the same, in which case you could have subtraction from background. You could actually use hormetic effects, I believe, through a post-conditioning mechanism to slow down the rates of aging. And so it worked post-conditionally instead of preconditionally. And also there was a guy I saw on YouTube, he's from the University of Toronto, and he was studying shock. He was inducing shock in mice and rats by removing blood and doing other things. And he took the cuff and put it on its hind leg, and he did the same routine, and then he induced massive shock. And then the control groups didn't get that. They had lots of damage to their lung, their liver, and their kidneys. Pretty striking. Those that got the cuff, they were reduced by about 80% in damage. That resonated with me because 5 years ago, 8 years ago now, I guess, I finished a 50-mile bike ride. I was just about to get home on a Sunday afternoon, and I fell off my bike and essentially broke my pelvis in 2 places. It actually wasn't too much fun. And so I got this big line of black and blue down my right leg, and I said to the doctor, where'd that come from? He said, well, you had tons of internal bleeding. He said, you were lucky that I fell down, you won't believe this, right next to the fire station. And it had an ambulance. It was Sunday afternoon. They were probably watching the Patriots play a football game, and they had to come out and take me to the hospital. And so he said, but if you fell down out in the middle of cow country that you normally ride, you could have just bled out there. He said, and so really? And then I saw this guy in Toronto with shock in his thing, and I said, me and my bike riding, maybe I should be, essentially pumping up my arm before I go out for the bike ride. And so I had to invite him to speak at one of our conferences. And so it was actually pretty good. So now, in the world of, there's a lot of application that you can find, and I started this presentation off by talking about that it kind of compares with how do you compare with the threshold and the linear model. Well, here I'm going to try to tie it all together for you, and this is what I would recommend to regulators. Okay, this is the linear model, and this is a hormetic little dip, and then I don't have it quite represented here, but a threshold. It's a very interesting thing. At the nadir of the hormetic curve, in general, I find that actually corresponds, if you were to believe in a linear model, that corresponds to a risk of about 1 in 10,000 cancers. But I'm saying I don't believe in that linear model. I believe in a hormetic model, and that shows a drop in cancer risk by 30% or so. If you assume that both of these 2 models are wrong and used a threshold model with a couple of safety factors, you'd come out almost exactly at this right thing. At this point, the 3 models line up. It actually was pretty amazing to me. So I called it the regulatory sweet spot, and expected everybody to say how brilliant it was, and they were going to change the regulation to accommodate my views, and of course that hasn't happened. So I'll give you some conclusions. In my opinion, hormesis is a general and central biological concept. It affects all disciplines utilizing the dose-response concept. It represents a general adaptive strategy through which biological performance is enhanced and mediated. Hormesis provides the theoretical basis and the molecular foundations to create biological shields. Really, how to protect yourself, actually, against chemical, radiological, and microbial threats, and I believe psychological threats as well. Whether you're going into, I was really impressed with the preconditioning going into surgery with the ozone. Now, all those ozone preconditioning applications said, wow, he is really got it. So I was excited about that, and it fit in nicely to my thinking. This has implications for numerous and diverse medical interventions, as well as affecting the health and performance of military personnel, athletes, football players who get concussed. They could be preconditioned every week before a game. Accident victims and so forth. And failure to consider hormesis within hazard assessment and risk assessment is a serious failing of modern risk assessment. And risk assessment needs to consider the entire dose-response continuum in order to serve the public health. Thank you very much.